So I think we are all in the problem of cardiovascular disorders already, and actually Fabio talked about the possibility to induce myocardial regeneration, which is one of the ways to cure, let's say, a myocardial infarction. However, even if you are able to induce cardiomyocyte proliferation and regeneration of the muscle tissue, it is a common experience that the heart is not able to spontaneously revascularize itself, and this is why after a myocardial infarction, the tissue is going to die. So if you induce the proliferation of cardiomyocytes, but if you don't restore blood perfusion, actually you're not going to cure the infarct. And in addition, a lot of patients are not suitable to the surgical commonly used revascularization procedure. So there is a plethora of patients that would benefit for the possibility to induce new blood vessel formation in ischemic tissue. And a lot of clinical trials have been attempted trying to deliver pro-angiogenic molecules, so molecules able to induce the formation of new blood vessels. First among all, the vascular endothelial growth factor, as you will see, and these trials have invariably failed. So when clinical therapeutic benefit, zero. Why? It is well known, and Fabio already introduced you to the concept that at birth something happens in our heart which blocks any kind of regenerative process. So during fetal development, cardiomyocytes proliferate, there is a lot of angiogenesis ongoing, and something happens at birth that stops the regenerative potential of probably all the cells in the heart. Cardiomyocytes completely change their shape, their metabolism, their contractile property, and their regenerative potential. And over the years, we have been investigating a lot of possible mechanisms and events that occur at birth that might be responsible for the loss of this proliferative potential. So at birth, the heart suddenly experiences a mechanical stretch because there is an expansion of the blood volume in the heart. There is a sudden increase in the oxygen levels, and hyperoxygenation and production of oxygen-free radicals have been implicated in the DNA damage and loss of proliferative potential of cardiomyocytes. We know that there is a shift from glycolysis to fatty acid oxidation, and also at birth the heart, as many other tissues, however, are suddenly not anymore exposed to circulating factors from the maternal blood that might be responsible for this differential proliferative potential. So we reason that the same mechanism that are implicated in stopping cardiomyocyte proliferation could also stop the proliferation of endothelial cells, thereby inhibiting angiogenesis in the adult heart. So going back to the process of new blood vessel formation, over the years we have elucidated the molecular players that are involved in the formation of a new blood vessel, which is called therapeutic angiogenesis. And most of these information have become available from the study of knockout animals. The circulatory system, the heart, and the blood vessels is the first organ that forms in the embryo. And schematically we can recognize at least 3 mechanisms that are responsible for new blood vessel formation. First one is called vasculogenesis, and this is what happens in the embryo, in which in a few regions these structures appear that are called blood islands, composed by specialized cells that are the angioblasts. And Angioblast or hemangioblast give rise to both endothelial cells that line the blood vessels and also to hematopoietic cells that populate the blood. Subsequently, this primitive plexus has to undergoing a maturation phase in which extracellular matrix and smooth muscle cells, pericytes, are recruited to cover these newly formed vascular tubes to form mature vessels. This only happens in embryos, although some works suggested that vasculogenesis-like processes can also occur in adulthood, but this has been harshly questioned. Probably it's not true. So adult mammals have two alternative mechanisms to form new blood vessels. First one is angiogenesis, which is a process that starts from immature vessels, in which part of its vessels get destabilized, and endothelial cells, in response to pro-angiogenic stimuli, start proliferating, migrating, and forming sprouting structures, new sprouting tubes that originate from a pre-existing vessel. And this is the most common mechanism that form new blood vessels in cancer, in eye neovascularization, during the female cycle, the estrous cycle. So wherever we have skin wound healing and so on, wherever we have new vascularization in adults. There is a third mechanism that is called arteriogenesis, which is essentially a response that is most commonly occurs during chronic ischemia, and this consists in the enlargement and restructure of small vessels, small capillaries, small arterioles that are not patent or purely perfused in normal conditions. In response to chronic ischemia, they can become larger and become more resistant vessels, and this is called arteriogenesis. So as mentioned, the circulatory system is the first functional unit in the developing embryo, and it occurs with very similar mechanisms in mice and humans. So what we know about the molecules involved in the formation of the in the determination of angioblast and subsequently in the formation of the primitive vascular plexus, as I said before, comes from studying from knockout animals. And this is one example, one of the first studies that show that in the absence of this gene, FLIC1, mice don't develop. This knockout is early lethal, and it has major vascular defects, so blonde islands don't form. What is this FLIC1? It's the receptor, the major receptor, is also called VEGF receptor 2, because it binds the major angiogenic factor, that is the vascular endothelial growth factor. And indeed, the knockout for VEGF has a phenotype which is very similar to the one induced by the knockout of the receptor, because they have to bind each other in order to develop the in order to promote the development of blood islands and subsequent formation of the vascular plexus. And these knockout studies also indicated that, as we mentioned before, VEGF gene has to be finely tuned in terms of dosage, because even heterozygous mice have a lethal phenotype. So it is really important that the VEGF is expressed at the appropriate dose. And so numerous knockout mice clearly indicated that angiogenesis depends on the proper interaction between a number of proangiogenic molecules, and this is a schematic representation of various members of the VEGF family of factors that interact with a family of receptors that are indicated by progressive numbers. So the main pro-angiogenic molecules is VEGF receptor 2 or FLIC1 in mice, because this induces a signaling cascade that overall induces proliferation, migration, and survival of endothelial cells, which are all required to form new blood vessels. VEGF receptor 3 is more involved in lymph-angiogenesis, so proliferation of lymphatic endothelial cells. And receptor 1, I will mention it later, it is considered an anti-angiogenic molecule because it acts as a trap for some of the VEGF members, not allowing them to interact with VEGF receptor 2. So its role is debated, it has a role in monocytes, but overall it has an anti-angiogenic function. And so the numerous knockouts that have developed have clearly shown that VEGF is the main inducer of angiogenesis, but then the primitive vascular plexus has to mature in order to form functional blood vessels. And there is a number of additional molecules, angiopoietins, that have to interact with type receptors in order to recruit peri-endothelial cells, pericytes, smooth muscle cells, and stabilize the vessels, or PDGF, TGF-beta, that are responsible for the remodeling of the plexus to form a structured network of arteries and veins. The whole system is triggered by the lack of oxygen. This is a common experience. Even tumors induce neo-angiogenesis when they get hypoxic. So what happens is that low level of oxygen through molecular sensors that are the PHDs proteins stabilize the transcription factor that is HIF, hypoxia inducible factor 1, and these in turn induce the secretion of vascular endothelial factors and transcription of other genes that induce glycolysis, so all genes that are necessary for the proper proliferation and migration of endothelial cells. Normally, HIF1 is ubiquitinated by this ubiquitin ligase, that is the VHL ligase, the gene of which is, when mutated, is responsible of the von Hippel-Lindau syndrome, and indeed in this syndrome in which this ubiquitin ligase is inactive, HIF1 is constantly stable because it is not ubiquitinated, not degraded. So these people have a high risk of formation of hemangioblastoma because they have an ongoing angiogenic signal constantly active, as if their tissue are constantly hypoxic. And this is the kind of hemangioblastoma that are formed particularly in the cerebellum and in the central nervous system. Nobody knows why exactly this preferentially happens there. Another important information in understanding the mechanism that lead to new blood vessel formation, and this has emerged over the more recent year, is that endothelial cells are not all the same, they need to be specialized. And this specialization, one of this specialization type discriminate between the endothelial tip cell, which is the cell that leads the sprout, and is the one that respond to chemoattractive stimuli such as VEGF, but also other stimuli that redirect the directionality of growth. If you think in our body, vessels and nerves often grow and line parallel in vascular nervous bundles. So it is not surprising that the same molecules, such as natrins, for instance, that direct the directionality of axonal growth cone, are also able to direct the navigation of endothelial cells, because nerves and vessels share a lot of mechanisms, and indeed they often develop along the same route in the body. So these cells is very sensitive with these filopodia, lamellipodia to chemoattractive and chemorepulsive cues, whereas endothelial cells laying behind the tip cells are those that actively proliferate and form the elongated vascular sprout. and we are starting understanding the mechanism beyond this specialization. So we know that a high level of VEGF, as in hypoxic condition, actually start acting on a cell and transform this cell in a tip cell. So the first cell that respond to VEGF become a tip cells. It upregulates the DLL4, and through the notch signaling, it downregulates VEGF receptor 2 on the adjacent cells that thereby become stalk cell and can proliferate actively. So this is more or less the same concept. And then another event that needed to happen is the specification of endothelial cells to be either an arterial or a venous cells. This is something that originally was postulated to be induced by hemodynamic forces because the flow is different in arteries and veins, whereas again, genetic evidence and knockout animals clearly indicated that arterial-venous specification is already determined before the onset of blood flow. So there is a genetic program that directs the specialization of these cells to be either arterial or venous. We know now various markers. For instance, arterial vessel are positive for FRINB4, venous endothelial cell are positive for FRINB2, venous cells are positive for FB4 receptor. We know that these cells have to interact at the level of capillary fusion, and this induce functional specialization, and a lot of additional markers have been identified. So arteries are usually positive for FRINB2, express high level of DLL4, and this is why they don't branch so much, because DLL4 interact with notch, inhibiting tip cell sprouting, and they are also highly positive for neuropilin 1, whereas venous endothelial cells are positive for the FP4 receptor and have high level of this transcription factor. So, going back to our starting slides, now that we know more or less the molecules that are involved in the various phases of angiogenesis, how can we play with them in order to induce functional formation of new blood vessels? So as I showed you before, AAV vectors are very powerful in transducing any kind of muscle tissue, skeletal muscle or cardiac muscle. This is the angiogenic response you obtain if you deliver the VEGF-A, the most potent angiogenic molecules, in a skeletal muscle. This is a normal muscle with the normal number of arteries here in brown. And this is after 1 month of VEGF overexpression. You have a huge increase in the number of arterial vessels. However, if you perfuse this vessel with a fluorescent dye, you clearly see that in a normal muscle the dye stays intravascular. These black holes are the muscle fibers, and these green lines around each fiber are the capillaries that surround each muscle fiber. If you overexpress VEGF, the dye extravasate, because VEGF is known to induce permeable, not functional vessel. And so over the year we have tried various strategies to induce the maturation of these vessels. One is to co-deliver factors that are playing a role in the later phases of maturation of blood vessels, such as angiopoietin 1, I mentioned it before. And indeed, if you co-inject VEGF with angiopoietin 1, again, exploiting the property of AAV vectors to enter together into the same target cells, you can co-deliver cocktails of therapeutic genes. Normally, VEGF induces permeability. Here is another way of measuring permeability, measuring the blue dye that extravasates. If you co-inject it with angiopoietin 1, this permeability is importantly reduced. And this results in a proper function of the newly formed blood vessel. This is something that we perform also in PISA. Fabio before mentioned the possibility to use a PET scanner to evaluate function in dogs and pigs, but actually we had the chance to try to use it in rodents as well, and somehow it works. You don't have a huge resolution, but this picture, for instance, show you that you can perfuse animals with a radioactive molecule that shows you perfusion with this color scale. And you see here that actually if you inject VEGF in a leg compared to the other leg, you don't have a huge increase in the perfusion of the VEGF-treated leg, despite that huge number of arteries that we saw before. And if you induce the animal exercising, as you can do, for instance, if you connect a pacemaker to the leg in order to stimulate muscle contraction, you see that most of the organs undergo the normal vasodilation that you have during physical exercise, the brain, the heart, the kidney. The contralateral legs also get more perfused. The one injected with VEGF is less perfused because VEGF induces this huge extravasation of blood that eventually is negative for the effective muscle perfusion. However, if you co-inject VEGF with angiopoietin-1, perfusion increases. And another way, we were mentioning this strategy before, to regulate and control the maturation of blood vessel is to control the dose of VEGF that you have. We know that excessive VEGF induces excessive endothelial cell proliferation and permeability and immature vessels. So we can develop strategies to control the amount of VEGF that it is produced using promoters that are responsible to, for instance, this Tet repressor. This is a molecule that normally binds to this promoter and switch expression off. However, if you administer doxycycline, a drug, this repressor moves away from the promoter and allows transcription of VEGF gene. So normally, if you co-transduce a target cells with the repressor and this inducible construct, you have expression off because the repressor is constantly bound to the promoter. If you administer doxycycline, you remove the repressor, you activate transcription. This works in cells. No doxy, no VEGF, you give doxy, VEGF starts to be produced, you remove doxycycline, VEGF expression is switched off, and the same happens in vivo in mice. You can give the doxycycline to the drinking water of these mice, you start administering doxycycline, you activate VEGF expression in a muscle, you remove the doxycycline from the water, VEGF is switched off, you give doxycycline again, it activates transcription again. So what happens to the vessels? Here you see again what I showed you before, normal vasculature, VEGF-induced vasculature after 30 days of VEGF expression. However, if you leave VEGF to be expressed for 30 days and then you switch expression off, you allow this vascular structure to mature and you end up, you see better here, normally in an 80 micron section you have 3 capillaries around each muscle fibers. Here you cannot count because everything is fuzzy. But after 30 days of VEGF expression and additional 30 days off, you end up with 8 capillaries in the same thickness. So an increased number of vessels that have a more or less normal shape and function. So here again we took advantage of SPECT to show that this strategy actively induces effective neovascularization. Then in collaboration with Professor Recchia, we moved to large animals, trying to get this into a therapy. And we established a model of myocardial infarction in dogs. As Fabio mentioned before, you can chronically instrument this dog to occlude the coronary artery and then have a measure of local contractility, adding pairs of piezoelectric crystals at the site of the infarction and in a remote region. And monitoring the movement of this piezoelectric crystal by echocardiography, you can have an idea of how much the heart contracts. So this paradoxical movement after the infarction means that during systole, when the heart contracts, the 2 piezoelectric crystals in the infra region that cannot contract gets away from each other because during systole the infra bulges out, so the 2 crystals get away from each other. And this paradoxical movement in the control heart is progressively reabsorbed, and at 1 month you don't have any movement because the scar becomes completely akinetic. In VEGF-treated animals, you see that we could recover up to 80% of contractility. And what was most interesting to us was that the two cures started to diverge very early after VEGF delivery, 48 hours, which is a time not compatible with the formation of new blood vessels. So to make a long story short, this led us to understand that this protective effect that we saw by VEGF was not due to the formation of new blood vessels, but to the fact that cardiomyocytes express VEGF receptor 1, and probably this protective activity was due to a kind of reprogramming of the metabolic activity of cardiomyocytes, rendering them more resistant to hypoxia. At the same time, we could observe that actually if you see the effect of VEGF in the heart, this is very different from what I showed you before in the skeletal muscle. So if you deliver VEGF to the skeletal muscle, you have a huge increase in the number of endothelial cells that actively proliferate. They even form this structure that are kind of angioma-like, the kind of lesions that I show you in the von Hippel-Lindau syndrome that replace the muscle fibers. So you have completely messed up of the muscle structure because endothelial cells are very active. They form a lot of capillaries and arteries. In the heart, you do see a small increase in the number of arterial vessels, but no endothelial cell proliferation, despite same level of AAV vector there and level of VEGF expression. And so this suggests that something is there in the heart that don't allow it to respond properly to VEGF. And so I quickly show you this model, and Nina in 2 days will tell you more about this model. We exploited this system that allows to assess in vivo the appearance of sprouting endothelial cells. So in this system, you have the Cre recombinase, tamoxifen inducible, under the control of the apelin promoter, which is a promoter active in the tip cell. Remember the cell that leads the front. So only tip cells express the Cre recombinase, and upon administration of tamoxifen in vivo, this switch red fluorescence into green fluorescence. So normally these mice have red fluorescent cells, and only apelin positive tip cells become green upon tamoxifen administration. We deliver VEGF in these mice, and we saw that both skeletal muscle and heart activate apelin expression in tip cells. So tip cells form, they do respond to VEGF in both organs. However, only in the skeletal muscle, green cells become incorporated into arterial vessels. In the heart, they don't. So we took these cells and endothelial cells out of these mice from skeletal muscle and cardiac muscle to assess their capacity to sprout in these spheroid systems. And again, you see that the number of green cells is more or less the same in the two spheroids, but endothelial cells from the skeletal muscle are able to sprout in response to VEGF. You see here this nice green tip cell that lead the sprout. From the cardiac muscle, they cannot sprout. And this is exactly what you see in vivo. Green cells form these elongated tubes in the skeletal muscle, whereas they stay as single cells in the heart, and we know that they eventually die by apoptosis. And Nina will show you more. We also did this interesting experiment. We took EGFP positive cells from embryo of EGFP mice in order to have very actively sprouting endothelial cells and genetically labeled by EGFP gene. So we have these green fluorescent endothelial cells that we injected either in the skeletal or cardiac muscle. And again, in the skeletal muscle, they contributed to the formation of new vessels. In the heart, they survive for a few days and then they disappear. So this suggests that something is there in the heart that block the capacity of endothelial cells to survive, proliferate, migrate, and whatever is needed to form new blood vessels. So what is there that blocks? In the lab, we are investigating various contributions from resident cells in the heart that could block the proliferative capacity and angiogenic capacity of endothelial cells. One of the resident cell type are cardiac fibroblasts. So we set up this co-culture system in which we culture endothelial cells, cardiac endothelial cells, with either embryonic fibroblasts and perform this scratch. And you see that embryonic fibroblasts start to migrate, and they seem to pull and attract also endothelial cells in red to close the gap. Whereas here, adult fibroblasts in the same co-culture system with the same endothelial cells migrate much faster compared to embryonic fibroblasts and don't allow the penetration of endothelial cells in the gap. Look at this endothelial cell, red. It's trying to migrate, but as soon as it meets this fibroblast, it retracts and go back. And we are now assessing the role of a few inhibitory cues, repulsive cues, semaphorins. We have these two candidates that might be responsible for this inhibitory effect by adult cardiac fibroblasts. Another cell type which is there and might be responsible of this inhibition of angiogenesis in the adult heart are cardiomyocytes. So again, we set up a co-culture system of cardiac endothelial cells and adult cardiomyocytes, and we observe that adult cardiomyocytes inhibit the proliferation of cardiac endothelial cells. And again, we have a candidate. Remember that I mentioned initially that receptor 1 is considered an anti-angiogenic molecule. This has been clearly established by this elegant paper by Ambati in this Nature paper in 2006, where it showed that the avascularity of our cornea is ensured by the production of abundant level of the soluble isoforms of VEGF receptor 1. That acts as a sort of sponge. It sticks to VEGF and it inhibits VEGF interaction with the proangiogenic VEGF receptor 2. So since we could demonstrate that VEGF receptor 1 and its soluble isoform is upregulated in the postnatal heart, we thought that maybe the same mechanism may exist in the adult heart. And using the CRISPR-Cas system, we silence FLT in cardiomyocytes, and indeed we saw that this partially restored the capacity of endothelial cell here to proliferate, incorporate EDU in vivo in response to VEGF. And finally, we are assessing another possibility. I mentioned in the initial slide that some additional events that occur at birth that might be responsible for this block of regenerative potential are mechanical forces, because the heart is exposed to an increase in volume load at birth. And so we moved to Innsbruck to set up this model of heterotopic heart transplantation, in which the heart is removed from the thorax and anastomized two vessels traditionally in the abdomen. Here they modified the protocol to have the heart implanted into a neck and anastomose in the aorta with the carotid artery and the pulmonary artery with the jugular vein. In this way, the heart is perfused by oxygenated blood as any other organ in the body, but the ventricles are completely unloaded because they are not filled by blood, and the heart doesn't have to face the pressure in its chamber. So in this unloaded heart, reduced mechanical forces, we could detect some extent of cardiomyocyte proliferation. PCM1 is a marker of nuclear cardiomyocyte nuclei, and you see here EDU incorporation at a very high extent, possibly higher than with the microRNA. Female proliferate more than mice, but this is another story. I don't have any insight in why this happens. Also, endothelial cells proliferate more in the unloaded heart. And even more impressive, if you deliver VEGF in this condition, you lead to the formation of this structure, which are very reminiscent to the angioma-like structure that I showed you in the muscle. It seems that endocardial cells, so the layer of endothelium in the lumen of the heart, sense VEGF and it invaginates, forming this effect that we call Venice lagoon, because it seems that endocardial cells start proliferating, forming this structure, leaving islands of these cardiomyocytes. And finally, just a final concept. Again, Nina will tell you more about this. We are trying to understand if and to what extent this poor angiogenic potential is correlated to the low incidence of cancer and metastasis in the heart. Again, it is a common experience that the heart rarely is affected by cancer and metastasis, despite most of the blood flows through it repeatedly. And again, we started injecting cancer cells into various organs. And again, where in the skeletal muscle we could see vessels penetrating the tumor mass, forming these vascular structures in the heart. Resident endothelial cells are not able to penetrate the tumor mass, and this results in a much reduced tumor growth compared to other organs. Here I show you the skeletal muscle, but the same holds true for other site. Tumors invariably grow much less in the heart. So with that, I think I finish. I just have to thank you, the people working with me in my group that made most of the experiments I show you, and thank you all of you for your attention.